Hello, biology world. Purpose of this video is to give students that were in this Zoom an opportunity to see the questions again and to practice with their study guide, practice with their notes, get ready for the quiz, but most importantly to give those students who are unable to be here an opportunity to see questions like those that you will see on the test. A good kind of rule of thumb, kind of an assumption you can make is that if you know the answers to a lot of the questions that you see in this video, the quiz should be markedly easier. Because remember, the quiz is multiple choice questions. You will actually see the answer. In this case, you will not see the answer. You will have to know what the answer is. So quiz is going to be a heck of a lot easier than this. So this will be a good gauge to tell how much preparation you need to do over the coming days. Okay, what I'm going to do is I'm going to give a certain period of time so I can be consistent and not have to set my timer a whole bunch. I'm going to do all the 1-point questions, and then these 2-point questions, and then these 2-point questions, and then these ones. And again, for those participants that are in the room, if you wouldn't mind just putting your hand up when you know most of the answers that you see or all of the answers that you see, or you're pretty sure you know what the answer is. Put your hand up, and when most people have their hand up, we will move on. Okay, here we go. Any questions for me, participants, before we get started? All right, best resources are your study guide, okay, and your class notes. And depending on how good of a reading note taker you are, potentially your reading notes. And I certainly would have the book open as well. All right, here we go. Okay, true or false? Genes contain instructions for making proteins. And again, for those of you that have your hands up or you already know what the answer is, just be patient, because it's possible that you have prepared a little bit more than others that are in the room. And we just simply might have some people who don't feel like raising their hand, which is totally fine. I'm not going to make you guys raise your hand. About 10 more seconds. All right. Okay. Correct answer, of course, is true. Remember, genes are segments, pieces, specific pieces of DNA that will code for messenger RNA. And then that code is taken out of the nucleus by messenger RNA in the form of 3-letter sequences called codons. And then, of course, at the ribosome, that codon matches up with transfer RNA's anticodon. Each codon for messenger RNA specifies a specific anticodon for transfer RNA. And of course, that transfer RNA is going to be carrying a very specific amino acid. So translation, which is what happens at the ribosome, is translating DNA's code through messenger RNA into proteins. Do you have a question, Ala? Okay, just forgot your hand up. All right, you guys, moving on. Okay, true or false? Proteins, polypeptides, and enzymes are all made by constructing long chains of amino acids. So basically, this is a true or false question that proteins, polypeptides, and enzymes are all kind of the same thing. That's really what this question is saying. All right, here we go. That, of course, is true. Polypeptides basically are kind of the raw protein, if you will. They start being made at the start codon, they stop being made at the stop codon. And then, as you guys heard in the video, the crash course video, some of those molecules in the amino acids, bend to bind to other ones. Some of them are hydrophobic, meaning they don't like water. Some are hydrophilic, which means they don't mind being on the outside. So folding takes place to actually produce the protein. And of course, remember back to last semester, an enzyme is a very unique type of protein that does things for the cell. So a polypeptide, a protein, and an enzyme, they're all made of amino acids. All right. Did we do this one? Yeah. Oh my gosh, it's in there twice. Weird. Okay, here we go. Blanks are always changes to genetic material being copied. What are those changes to genetic material? Again, hand up if you think you know the answer, or you know the answer. All right, here we go. So any change to DNA, okay, which is the original genetic material, it's the instructions for life, is referred to as a mutation. All right, here we go. Which enzyme is responsible for making messenger RNA and for checking to see if the sequence is correct? So it's got a proofreading function as well. When looking at the wheel on page 303, what do those three-letter combinations represent? And be as specific as you can for that second question. Again, hand up if you think you know both those answers. All right, here we go. Okay, that enzyme is referred to as RNA polymerase, just like DNA polymerase built the new DNA strands in DNA replication and then proofreaded it. RNA polymerase functions the same way when messenger RNA is being built inside the nucleus. Of course, we call that process transcription. So RNA polymerase. And then when you are reading the wheel inside out in those three-letter sequences, those are mRNA codons. Okay, messenger RNA codons. And remember, the lab that we did last week, okay, had you use transfer RNA anticodons to figure out what the amino acids are. That is the only time we will ever use transfer RNA to figure out amino acids is in that lab. Everything else, you're focusing on the messenger RNA codons. Samarin, did you have a question? Okay, here we go, folks. Okay, during blank, a protein is formed from the joining of amino acids in the cytoplasm at a ribosome when transfer RNA brings those amino acids based off of whose codons. Okay, so what's the process called? And whose codons are being used to determine transfer RNA and the amino acid? How many bases are needed to specify 5 amino acids? All right, about 30 more seconds. Okay, here we go. Okay, so that process of making proteins, the scientific word for protein synthesis is translation because, again, DNA's code through messenger RNA is being translated into proteins with the building of amino acids. Okay, translations, and for every base, or pardon me, for every amino acid, that's a mistake. My bad. Hold on a second. Whoa, hold on. Should be 15. Whoops. All right. Sorry about that. Yeah, so for every amino acid, it's 3 bases. So however many amino acids you see in the test question, multiply it by 3, and that is the number of bases. Because remember, each codon has 3 bases. All right, let me get my participant list back. Okay. Do you have a question, Lucas? Lucas? No problem. All right. Yeah, you guys don't have to tell me, no, I don't have a question. Just simply take your hand down. All right, here we go. All right. During transcription, RNA is formed where? How many types of RNA are there? Give about 30 seconds. All right, you guys, here we go. Okay, so transcription, remember to transcribe means to copy. All right, so to transcribe and the copying of DNA takes place in the nucleus, and they're the three sisters, Myrna, the busy one, Turna, yeah, sometimes, and Erna, yeah, she's lazy. Okay. Blank mutations can potentially change every amino acid after that point. True or false? An inversion mutation is a type of gene mutation. And on that true or false question, be very careful. Look at the spelling. That's not meant to be a trick question, but it could be if you're not careful. Yeah, go ahead.
So for the additions or deletions, it removes like a single letter in the genetic code and moves everything over one, right?
Correct. Well, yeah, moves it. It moves it to the right if it's an insertion. It moves it to the left if it's a deletion. But the bottom line is, all you need to remember is that changes every single codon after that point, which means that it could change every single amino acid after that point. Yeah, so it goes to the right, it shifts it over if a letter's added during an insertion, and it moves it to the left if one is removed during a deletion.
But if they're all moved over, then there's like one missing at the very end. There'll be one extra.
Yeah, I'm not really sure how the cell deals with that extra letter. I mean, if it doesn't actually ever get to a stop codon, I mean, I would think that it just keeps going, but hundreds of millions of years of evolution, I would think that a remedy for that has somehow evolved. So I'm not sure exactly how it ends if that's the case, but logically, it could just continue making that protein until the enzyme dies. All right, here we go. Here are the answers, guys. Okay, so a frame shift, and there are two types of frame shift, insertion and deletion. And of course, an inversion is a chromosomal mutation, not a gene mutation. An insertion, okay, which is spelled a little bit differently, it'd be an S and a T. An insertion deletion would be a gene mutation. So, of course, that is false. Okay, unlike DNA's thymine, RNA has what? If DNA has a sequence of TAT, GCA, TGA, what will the messenger RNA codons be? So a little base pairing for the second one. And be really careful whenever you see DNA and RNA in the same question. Well, honestly, the only time you would see DNA and RNA in the same question, well, RNA in the same question, it would be messenger RNA. Remember, there are no T's in RNA, ever. No T's are ever found in RNA. And that's what the first part of the question is about. So whenever you're using DNA to make messenger RNA, get rid of the T's and replace them with this thing. All right, you guys, here you go. Okay, instead of thymine, RNA has uracil, so that means A would go with U. Whenever you're talking transfer RNA, messenger RNA, A goes with U, U goes with A. The GC combination is still accurate. And if DNA has a sequence of TAT, so T normally goes with A, so that's going to be A. A now goes with U, AUA, C, G, U, A, C, U. Okay? Most common mistake students will make is they'll have a T here, they'll have a T here, and they'll have a T here because they forgot that whenever you go to RNA, you got to replace the T's with the uracil right here. All right, which type or types of RNA are involved in protein synthesis? Remember, protein synthesis, start to finish, copying down DNA's code all the way to the end of the polypeptide chain. That entire process is protein synthesis. If messenger RNA has a codon of UAC, what will transfer RNA's anticodon be, and what will the amino acid be? And remember, be careful. We don't use transfer RNA to figure out the amino acid. You use the wheel, and then you use the codon. All right, here we go. Okay, so protein synthesis, start to finish. Messenger RNA gets DNA's code. Transfer RNA carries the amino acid. Ribosomal RNA makes up part of the ribosome. All three are related to protein synthesis. And of course, transfer RNA is going to be U goes with A goes with U, C goes with G. And if you use UAC in the wheel, you will get tyrosine. And you can always abbreviate tyrosine as TYR. Well, actually, you don't need to worry about that anymore because you're just going to be doing A, B, C, and D on your test. But you can always do the first three letters of the amino acid unless we're talking about glutamic acid or aspartic acid. And then you have to do GLU acid or ASP acid. So remember that for AP Bio in a couple of years because some of you guys will be taking AP Bio, I suspect. All right, here we go. Which type of RNA functions as a blueprint of the genetic code? Remember, the genetic code is DNA. And what is produced during transcription. Be as specific as you can for that second question. All right, here we go. Okay, so messenger RNA is the blueprint of the genetic code. It is transcribed, it is copied in the nucleus, or DNA is copied. It is created by copying DNA. And of course, transcription is messenger RNA. Okay, which type of gene mutation will never be considered a frame shift? And what type of mutation involves one or a few nucleotides only? And this was today's and yesterday's warm-up questions. Remember, always have your warm-up questions right next to you when you're taking the test. About 10 more seconds. All right, here we go. Okay, so a substitution mutation is not considered a frame shift mutation because it's just replacing one letter with another one. It's possible, it's probable that the codon will be different, all right? But remember, because there are 64 possible combinations of codons and only 20 amino acids, it's possible that that different codon will still code for the same amino acid. Or if it doesn't, that the protein will still be able to work even with one amino acid different. Okay, so that's why frame shift mutations aren't as damaging or as dramatic as I termed it in the notes. And a point mutation involves one or a few nucleotides only. And of course, the three types of point mutations are insertion, deletion, and substitution. Okay, on to the 3-point questions. All right, here we go. 3 similarities between DNA and RNA, 3 differences between DNA and RNA. And remember, in the 12-2 class notes that we did at the end of last semester, we did cover the similarities and differences of DNA and RNA. And of course, you can get that out of section 12.2 in the book and section 12.3 in the book. Give about 10 seconds. All right, here we go. Okay. Differences. DNA has deoxyribose, RNA has ribose, DNA is double-stranded, RNA is single-stranded, DNA has thymine, RNA is uracil, DNA is in the nucleus, RNA can be inside and outside the nucleus. That's only the differences, though. My bad. Hold on, I want to make this. Hold on. Perfect. I forgot the Sims. Don't forget the Sims. Sims both have nucleotides, both have a sugar and phosphate group, both have A or C, G, A, both carry genetic information. That's good enough. Sorry about that, guys. My bad. Okay, let's save it. Okay. All right, here we go. Do you have a question, Nick? No problem. All right, here we go. What is the name for a 3-letter sequence in transfer RNA? Because there are 64 possible sequences of codons in messenger RNA, this means that each blank will have more than one messenger RNA blank coding for it. True or false? Spindle fibers are involved in protein synthesis. All right, you guys, about 30 seconds. All right, here we go. Okay, so the name of a 3-letter sequence in transfer RNA is the anticodon, which is complementary to messenger RNA's codon. I'll complete the sentence. Because there are 64 possible sequences of codons in messenger RNA, this means that each amino acid will have more than one messenger RNA codon coding for it. And you can see that if you look at the wheel. Like, for example, arginine has 6 different codons that code for it. And true or false, spindle fibers are involved in protein synthesis. That is false. Did anybody look up spindle fibers, and do they remember what process they are involved in? Anybody do that? It's actually what we're going to be talking about starting next week. It's called cell reproduction. Mitosis is the process where body cells or somatic cells replicate. And meiosis is the process where sex cells, gametes, egg cells for females, sperm cells for males, replicate. And spindle fibers are involved in pulling the chromosomes, the duplicate chromosomes, away from one another before the cell pinches. And you go from one cell, the parent cell, to 2 cells, the daughter cell. So spindle fibers are not involved in protein synthesis, but they are involved in cell reproduction or cell replication or cell division. They all mean the same thing. All right, here we go. Okay, genes contain instructions for assembling what? Given the following DNA sequences, what are the corresponding messenger RNA sequences? So again, these are DNA, and we know they're DNA because they have a T in there. So what would base pairing give you for this? And then use the wheel to determine what amino acids are coded for by the codons GGU and CUG. Move this over here so you guys can see the wheel. Obviously, the wheel is also on page 303. You don't need to use this copy. Okay, guys, about 30 seconds. All right, here we go. Okay, so genes contain instructions for assembling or making proteins or synthesizing proteins. Given DNA sequence, what are the corresponding messenger RNA? So A, G, C. A goes with U, G goes with C goes with G. T goes with A goes with U, G goes with C. A goes with U, C goes with G, C goes with G. And then we're using GGU. GGU is glycine, and CUG is leucine. So remember, you use messenger RNA to figure out the amino acids, and you read from the inside out. All right, last one. Okay. Which type of mutation is shown below? This is the original DNA strand. This is the strand that's being built. This is the original DNA strand. This is the strand that's being built. So what type of mutation for situation number 1? What type of mutation for situation number 2? Okay, here we go. All right, you guys. So the easiest way to figure this out is if everything else matches up, but you're one letter short, that means you have a deletion. If everything matches up, but you are one letter over, that means you had an insertion. And then, of course, you can take the next step and figure out where did the deletion take place. So in this case, this thymine was deleted. So instead of being TTC, if you remove that, it's going to be TCT, as you have there. And then it's going to be ACG, which you have there. And then it's going to be TA, and then you're missing one. Okay. Same thing down here. GGC, that matches up. That matches up. Whoa, the G is missing. So that means something was added here, which means GCA is now GCA. Okay. AAC is now AAA because it's reading these three. And then you have an additional C. All right. Okay, you guys, that is it for the questions. Let me get my video stopped real quick.